Hello, my name is Mike Ward. I'm the head of content for Informa Pharma Intelligence. We're here in Vienna at the Bio-Europe Spring Meeting where pharmaceutical companies, biotech companies, investors all get together to look at the industry trends, sort of seeing what's hot, what's not, where there are opportunities both for partnering and investment. One of the areas which has always remained an important unmet medical need, and there is still some interest in, is in the infectious diseases area, and specifically around the issues around antimicrobial resistance, where the sort of mortality rates associated with such infections is still very, very high, even in developed countries. So there's a lot of interest in sort of looking for new solutions. I'm joined by Graham Dixon, who's the CEO of Neem Biotech, which is a company that's based in Wales, and you guys have got a focus on sort of antimicrobial resistance. So could you describe the approach that you're taking?
Yes.
Because lots of people are there, but it's...
Yeah. Well, we believe we have a slightly different approach. We're coming at it from a non-traditional antibiotic angle. We actually have compounds that inhibit bacterial virulence and prevents them from becoming pathogenic and therefore becoming a problem.
Yeah. So, and how does that work? So, at what point is the molecule sort of impacting the bug?
Okay, so we hit a process called quorum sensing, which is essentially the way bacteria communicate with each other.
Right.
That then drives the production of various factors that allow them to colonize tissue, to produce biofilms, to actually damage tissue, etc. So we're hitting it at a fairly early stage in that process, preventing the bacteria becoming a problem.
So, whereas others are actually killing the bacteria, you're just basically preventing it from, I guess, communicating and growing.
Yeah, and infecting. So we all have bacteria on our skin, in our mouths that are no harm. In fact, they can be beneficial.
Yeah.
It's only when they become invasive and infective that they become a problem.
Right.
So our approach is meant to prevent that transition from a commensal to a pathogen.
So, because one of the challenges with sort of a lot of antibiotics is the fact that they wipe out, the microbiome, and there are sort of beneficial bugs. So, and they normally go at the same time. But your approach would actually protect the beneficial bugs.
Yeah. So we preserve the normal microbiome, but we will prevent that transition from a good to a bad bacterium.
Yeah. So, what sort of proof of sort of principle of concept have you achieved so far?
Well, our molecules are actually vinyl disulfides. They're synthetic derivatives of an original natural product lead. We have synthesized them. We've got composition of matter patents filed. We've gone into preclinical models in wound infections and in respiratory infections, particularly focusing on cystic fibrosis. And we have proof of concepts in models in the wound area, showing polymicrobial infections, we can control them in skin. Yeah. And we've gone into infection models in mice, for example, respiratory infection models, where we show we can very nicely control the organisms in those situations as well.
So two questions. One, are there any limitations to the sort of the kinds of bugs that you could actually target? And secondly, in the sort of the initial phases, which bugs have you decided to focus your effort on?
Well, normally, normal thinking would be that approaching virulence would be a fairly narrow spectrum.
Yeah.
Our compounds actually hit multiple sites in this communication, in the quorum sensing. Therefore, it's high up the pathway as well, so therefore we hit gram-negative and gram-positive organisms, so the spectrum in theory is broad. Yeah. Our focus has been really on Pseudomonas aeruginosa and Staphylococcus aureus in the first instance, because those are prevalent in wound infections. They're also very key infections in the respiratory tract, particularly in cystic fibrosis patients.
And there's also a lot of sort of antimicrobial resistance associated with those bugs as well.
Absolutely. And the way we work, because we're not hitting an essential metabolic process that kills the bug, as you referred to earlier, the actual pressure to evolve resistance is much less, because the bugs won't die, they just can't invade. They can live happily as a commensal, so it's the resistance should not be as much of a problem as with conventional direct-acting antimicrobial agents.
So you basically kind of put them in detention.
That's correct. Yeah.
Right, yeah. So that's the proof of principle concept that you've got. So what is the next stage for Neem with these molecules?
Okay, so we've the wound program is slightly more advanced than our respiratory. We are just at the stage now we've selected our clinical candidate and a backup compound that's a different chemotype. We're just starting IND enabling studies with a view to be in the clinic in a wound indication in 12 to 18 months' time. The respiratory program is slightly behind. As I said, we aim to select that clinical candidate by mid-year, and then we would trigger the IND enabling studies there for the inhaled development.
Okay, so as I mentioned, there is already quite a bit of interest in the infectious diseases space again, also from charitable organisations or government agencies as well, because of the public health challenges associated with it. So, how is the company financed at the moment, and do you have the financial runway to be able to achieve your development goals?
Well, the simple answer to that is no. We're very fortunate. We have a very committed and dedicated family office that continues to support us. But as everybody's aware, the costs as you go into the clinic increase exponentially. So we are looking for co-investment to be able to achieve that and accelerate our programs at the rate we could go. We have been well supported by non-dilutive funding from various patient groups. So, for example, the CF Trust in the UK, from Innovate UK, from the Welsh Government have given grants to us for the wound programme. So we're fairly attractive in terms of non-dilutive funding as well.
And pharmaceutical partners, because again, I guess to actually end up with a product that you're going to actually put into, is this going into sort of ICUs, or is it?
No, the wound one probably not, obviously, but the respiratory infections could be in the ICU setting, yes. The compounds work, they have some effects as a monotherapy in preclinical models. We need to prove that clinically, obviously, but they do synergize very nicely with current classes of antibiotics used in both wound and in respiratory infections.
Right. But you're looking for pharmaceutical partners to...
Yes, right. We're looking for financing. That could be from venture capitalists, it could be from collaborations with bigger companies, etc. Yes, so we're open to various scenarios to bring in the money to allow us to develop our program.
Okay, well Graham, thanks very much for coming in and talking to us.
Thank you.
Cheers.
Bye.